Hello, this is Koby for Science Sapiens, and today we're talking about molecular genetics. So, before we can start talking about what DNA does, we need to know its structure, because everything in biology, all structures in biology relate to their function. So if it has a specific shape, it has a specific job, and DNA has a really specific shape that allows it to do its job really well. Alright, so first of all, what is DNA? We hear it thrown around all the time, but what it actually is it stands for something. It stands for deoxyribonucleic acid. And deoxyribo stands for deoxyribose, the sugar that's in DNA. Nucleic, and then nucleic acid is the type of molecule it is. So we'll see this in a little bit, but DNA is made of a sugar, a nucleic base, a nitrogenous base, a base made off of nitrogen, and then that's connected to a phosphate. And it's the basis for all of genetics. So like I said, it's composed of three main parts: a sugar, a phosphate, and a nitrogenous base. The sugar and the phosphate are the same for every nucleotide. A nucleotide, if you remember from the biochemistry video, is the monomer of the polymer, the macromolecule of nucleic acid. So lots of nucleotides add up to make a nucleic acid. And all nucleotides, all the pieces, have the same sugar-phosphate backbone. And we'll see this in the next slide, but I'm just going to draw it now. So basically it has a phosphate, and that's in green, right? And phosphate's just a chemical group, and that's connected to a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it would just be ribose. And then that sugar is connected to, let's not use green, this purple base. And the base will change depending on the nucleotide. But this right here, that's the sugar. And the phosphate, I'm just going to write as a P little I, is the same. It repeats for every single nucleotide. And when we put many of these together, we see that it forms something called the sugar-phosphate backbone. It's just this repeating structure over and over and over again. Okay? Talked about that. All right, so complementary bases is the idea that what I just drew is only half of DNA. There's a second half that connects, and it's in something called an anti-parallel structure. We'll get into that. But the bases, there's four main types of bases in DNA. There's adenine, thymine, guanine, and cytosine. Adenine only bonds to thymine, A only bonds with T, and guanine only bonds to cytosine, so G to C. And these, if you remember again from the biochemistry video, are connected by hydrogen bonds. So adenine and thymine have two hydrogen bonds that connect them, and guanine and cytosine have three. So that's why they only go together. So this means one segment of the DNA is the exact opposite of the other half. All right. And the idea with these halves is that one has to go up and the other one has to go down. And we'll see this in the next picture. And that's called anti-parallel. And that's the way they fit together. And then you've probably also heard this double helical structure, which basically, it's like a spiral staircase. So it's like taking a ladder and twisting the two ends until we get this nice spiral. That's the shape of DNA. All right, so here again is just a better picture than mine of DNA structure. So we can see again, we have this right here is the nitrogenous base, right? A with T, G with C. And we see 2 bonds between A and T and 3 between G and C. And then right here, this blue that I'm circling in yellow, that's the deoxyribose. That's the sugar. And each one of these points is a carbon. Okay? And that's just deoxyribose. It's a type of sugar, and it forms this very specific ring. And then connected to the sugar, what I'm circling in blue and they've drawn in yellow, is this phosphate group. And you see the phosphate from one nucleic acid connects to the sugar of the second. So this right here is one nucleic acid. Here would be a second, right? A third, right? And we see that we can kind of divide it down the middle, and we see there's two different halves. And one of them is upside down. And this is the anti-parallel structure. they run opposite to each other so they can bind. And then on one side, we see that this twists, and that's the double helix. Double because there's two sides, and helix because it's twisting, like a spiral staircase. Okay, so when cells divide, they need to copy all of their DNA and put it into the new cell. And that means they basically have to replicate. They have to double everything in the cell. And it's a very specific process, and it's called replication. So replication is the process of taking DNA and copying it. So you have two copies so that each daughter cell can have it. And this passes all the instructions. Like I said, genetic material controls the cell. So when we replicate it and pass it on, that's passing on all the instructions to the new cells. And it's called semi-conservative replication because if we look at our picture here, the blue is the original strand. So if I just have one cell, we've got a regular old DNA like that. And then we're going to unwind it, and we'll get into how that happens. We're unwinding it down here, right? Unwound. And this these red strands are the new DNA. And it's semi-conservative because if you think about this going all the way up until we've replicated the entire blue original strand of DNA, each new strand of DNA is going to have half of the old one and half the new one. So it's semi-conservative because we're conserving half of the original DNA for each new cell. And we're creating half of a new one. All right, so there's a bunch of proteins that work in replication. Proteins, remember, are these macromolecules that do all the work for the cell. Okay? So helicase is a protein that untwists DNA. DNA is in that double helical structure, remember, and we have to unwind it before we can get at it and start reading it and start copying it over. DNA polymerase is really important because it unwinds and copies the DNA. It does a lot of the work in this process. It goes through base by base, nucleotide by nucleotide, and pulls in the opposite and copies the DNA over. It also unzips, so it's detaching the two sides of the DNA when it goes along and does this. Ligase joins Okazaki fragments. We'll talk about that in a second. But basically, it joins little pieces of DNA together. It's kind of like a gluer. And then nuclease enzymes basically cut out mistakes. They perform excision repair. So when there's a mistake, which happens, everybody makes mistakes, so does DNA polymerase. These nuclease enzymes, will come and cut out the mistakes and replace them with new ones. And that's called excision repair. And then finally, we have telomerase. And telomerase is not that well understood, but what it does is it adds telomeres to the end of DNA. And telomeres are these segments at the end of chromosomes, long pieces of DNA, that protect. They're kind of like end caps, or if you can think about a shoelace, that little wax around the end, that's basically what a telomere is. And scientists believe that these telomeres have a lot to do with age and aging, because over time, when we copy DNA over and over again, we lose a little bit at the end of our telomere. And then telomerase adds it back in, but it doesn't get all of them. So our DNA gets shorter over the period of our life, and scientists think this has something to do with aging. So telomerase adds telomeres, these little ends that protect the DNA back on. All right, so there's a very specific process to replication. First, we have to untwist and unzip to create a replication bubble and a replication fork. So if we think about the drawing from before, this red is our initial strand, our parent strand. And we're adding these new blue strands. This right here is called the replication fork. And it looks just like a fork. We're taking the two strands and separating them. And then the replication bubble, because if we finish this drawing, eventually the DNA is going to come back together. This right here is the replication bubble. It's the area that's unzipped because we only unzip a little at a time. We only unzip a little at the time, so the replication bubble is this space that's unzipped at the time. All right, so we have the polymerase, the DNA polymerase that creates new strands going from 5' to 3'. What that means is just the direction. So if we go back a few slides. All right. So what we see is that the carbons have each of those points is a sugar, right? So we have numbers for labeling these sugars. Sorry, not these sugars. We have numbers for labeling these carbons. So each carbon, so this is our sugar, right? Here. Each of these carbons has a very specific number. So 5' is right here, is where the polymerase, or the phosphate connects, and 3' is where the next one connects. So if we go 5, 4, 3, 2 right here, that's the 3' carbon. So it's just a way of labeling. Again, we see it here, 5' up top, 3' on bottom. The other strand is going in the opposite direction, right? We have 5' at the bottom and 3' up top. So let's just go back to where we were. So that's just a way of saying, is it upside down or right side up? So we can only add new DNA in the opposite direction, right? We have to have that anti-parallel structure. So polymerase adds new strands only in the direction 5' to 3', which is 3' to 5' on the parent strand. So it's very specific. It only goes in one direction. And then we have something called the leading strand and the lagging strand. The leading strand constantly moves towards the replication fork, creating new pieces 5' to 3'. Very easy. The polymerase just goes right along. And we have something called the lagging strand. The lagging strand moves away from the fork, and because it's moving in 3' to 5' direction, it creates these things called Okazaki fragments. Basically what it does is it writes 5' to 3' pieces and then skips to the next place and starts writing another 5' to 3' piece. So this is kind of confusing. So let's look at this picture here. All right. So right here, what's this called? This is called the replication fork. And then this whole thing here is called the replication bubble. This whole thing here, right? Fork, and this whole thing here is the bubble. And we've seen we're just blowing up this picture right here. And we see that this right here is a leading strand, constantly moving towards the fork from the start of the replication towards the fork. And what it does is it copies, no problem, 5' to 3', right? This is the polymerase, comes right along, copies the whole thing in one straight line. Very easy. But what we see at the bottom here is we can't do that. We can only copy, remember, 5' to 3', which will be moving in this direction. But we're starting replication here, and we need to end it here. Excuse me. So what we do is we create these things called Okazaki fragments. So basically what we do is we copy from here to here, stop, then move back, copy from here to here, stop, move back, copy from here to here, stop, move back. And we just keep doing this over and over and over again. And these little fragments are called Okazaki fragments, sorry. And then we bind them with this ligase, right, glue them together. And again, DNA polymerase is still doing the copying. And it's just moving 5' to 3', and it has to keep going backwards. Keep doing these little strands, go forward, go backward. It's kind of like reading a book backwards, just reading one page at a time. So I'd start with the last page, read it, then I move to the second to last page, read it. That's kind of what's happening here with the DNA. And we see if this right here is called the point or the origin of replication, if this top parent strand has leading on one side, it means it has lagging on the other, and the same thing over here. And we're always moving, again, 5' to 3'. All right, so again, we have a very specific process. So DNA, there's 3 steps: initiation, elongation, and termination. So initiation, we have to start this process somehow, and that's done by an RNA primer, a really short piece of RNA that binds onto the very specific start sequence of the DNA, where we want to start replicating. The DNA polymerase recognizes this primer and binds on, and we start replication. It's then elongated, so the polymerase goes along and copies all of the DNA, and after that happens, or during that happening, it's proofread by polymerase, and repairs are made by the nuclease enzymes. And then eventually we'll get to the end, the polymerase will fall off. That's called termination, right? And then things like the telomeres will come and be added on by the telomerase. So again, 3 steps. We have initiation, elongation, termination. Initiation, RNA primer binds to beginning, DNA polymerase binds on, starts copying. Elongation, that DNA polymerase goes along. And then termination, it gets to the end, falls off. We get that whole new piece of DNA. And like I said earlier, it's semi-conservative. We have half of the parent strand and half of the daughter strand, half of a new strand on each of the 2 new segments of DNA. All right, so that is molecular genetics. That is the structure of DNA and how it replicates. Thank you guys for watching today, and remember, rate, subscribe, and leave any questions in the comments below. Thanks. Bye.